I got that right. Okay, so the first one is, again, a very simple definition. If you got it wrong, I'm not sure how to say, except you have to memorize it, put it on the next card, write it over and over again. But at this point, we've said that an allele is an alternate form of the gene when we covered pedigrees, when we covered Punnett squares, when we covered DNA. So just over and over again, I've been hitting you over the head. A gene is an allele is an alternate form of a gene. Just as an addition, what is a gene? It's a coding area. It's an area of the DNA that codes for gets transcribed. So good. If you didn't know that, that was on the protein synthesis test. You should know that, right? It's the area that they have a long 3-meter long DNA molecule, 2 molecules hydrogen bonded together. You got 23 pairs of them in your nucleus. There's a lot of DNA, but not everything gets transcribed. Not everything makes a protein. All right, so a gene is the part that makes a protein. Moving on. What is the probability that on 4 flips of a coin, heads will occur on 3 flips and tails on 1? So in order to figure that out, you have to figure out how many different ways could I get 3 heads and a tail. Well, there's 1, 2, 3, 4 ways I can do it. Obviously, each one of these is 50-50. But if you're dealing with genes, and maybe this one's one 3/4, and then this one's 1/4, then your fractions are going to be different. But in this case, your fractions are the same because it's either heads or tails. It's the only option. But there's 4 different ways you can get those. So it's 1/16 each. So the question is, you can only have one way. You can only get 3 heads and a tail one way. So it has to be, you're using the word or. You can only have this one, or this one, or this one, or this one. So because you're using or there, you have to decide to use the sum rule. So you're adding the 1/16 up, and you're gonna end up with 1/4 because it's 4/16, and that's equal to 1/4. Another way you can do it is you can say, look, I got four times. I have 3 heads, so that's heads h cubed, right? H times h times h is h cubed. There's four different versions of that, and then you're multiplying that times t. So again, 4 t's, a t here, a t here, a t here, and a t here. You multiply them all together, and again you end up with 1/4. So you can use the sum rule, and this is, I think, a little easier for me to visualize because I'm adding them. Or you can just turn this into an algebraic formula and then find the answer. Either way, you end up with the same answer, 1/4. Is everybody okay with that? That's tough. That's tough. It's a product rule. It's statistics, but you should know this. You should know this. Moving on, when we get into population genetics, we're going to come back to this, and you're going to hit it. You're going to see it applied. What would be the dimensions of a Punnett square when you have this cross? Well, I drew it here for you. It's a 2 by 2. Yes, bless you. 1, 2 by 1, 2. It's a 2 by 2 square. So that should be pretty straightforward. Everybody okay with that? All right. In dihybrid crosses, the ratio 9:3 to 3:1 indicates. Now, this is something that a lot of people got wrong. I want to emphasize that if you have codominance, you're not going to have a 9:3 to 3:1 ratio. That's the point of the ratio. If you have a 9:3 to 3:1 ratio, you know you don't have any one of the more complicated inheritance patterns. So this indicates that you have what you have is dominant recessive. And the other thing is that you're not dealing with any kind of adjusted assortment. So what that means is that when you have these genes on these chromosomes, remember the assortment happens during meiosis, and you're going to get which gene goes in here, or does it go in here? Does this gene go in here? Or does it go in here? So how they assort, what combination you get is random, and we call that independent assortment. In other words, these two don't go together, they just go independently. Do you remember mitosis and meiosis, and especially meiosis, right? When we're talking about meiosis, remember that you're separating, do you get mom's chromosome over here, or do you get dad's chromosome over here? Which version of the gene do you get in the gamete? And that's up for debate. That's because there's an independent assortment. They're not dependent on each other. They're intermediate dominance. We don't use that word, do we? Have we ever used intermediate dominance? No. So if you chose C, I'm questioning. I think you probably were just like, well, it sounds right, so I'm just going to grab it because I'm not sure what's going on. 3 alleles for each trait, again, is when you have that 3 alleles for each trait, codominance, you're not going to get a 93 to 3 to 1 ratio. We're talking about phenotypes here, okay? This is 93 to 3 to 1 is phenotypes. So codominance and 3 alleles for each trait, you're not going to have a 93 to 3 to 1 ratio. That's a sign that you have something more complicated than a simple dominant recessive relationship. And of course, you're talking about dihybrid cross. You're talking about two genes. All right. So in a cross, let me get rid of this. So when we're looking at in a cross of a round P with a true breeding round parent, so true breeding means what? And they tell you true breeding means it's homozygous. When they tell you true breeding, it's homozygous. So that language you have to know. You have to learn it. So they give you in the problem what the genotypes are. On the end of course exam, they might just tell you true breeding, and they expect that you know it's homozygous. Well, there's your homozygous, there's your true breeding, and this is round. So what do you think round is? After reading that, is round dominant or recessive? Dominant. Why did you say dominant? You're right. Okay, that's honest. You're reading this. She's right. Round is dominant, but how do you know that based on this? It says true breeding round parent. Which one of these two is true breeding?
The two capitals.
The two capitals, because they're the ones that are homozygous. So round must be the big W. Do you see the kind of logic you have to use? It's not hard, but it is tricky. You have to focus your mind. The minute you take your eyes off the ball, the minute you kind of just start to drift away, you're lost, and then you have to start all over again. Because if you miss just one little piece, you got the wrong answer. So this is big W is round, is the point. So when I saw this, and they asked what genotypic proportions would be observed in the offspring, well, that was easy, right? I just took this one, did a Punnett square. I see that 25% is going to be round. You have to figure out which one of these is round and which one is. Do you see that? You have to know, is it big W that's round? Is it big W that's round, or is it little w that's round, right? You have to know that. You have to figure it out from the problem, and the way you do it is you look and see true breeding round plant, that means homozygous. Which one of these two is homozygous? The second one here, right? The first one's heterozygous. Here's big W, little w. So when we're looking at homozygous, that means that big W must be round. Does that make sense? You have to work on your logic. These problems are all based on logic, on thinking straight. Deductive logic. So here we have the cross. You have 3/4 are round, and 1/4. Well, 100% is round. 100% round, right? Half of them are heterozygous. 1/4 are homozygous round, and 1/4. 3. What am I doing? I'm sorry. 50% homozygous dominant, and 50% heterozygous. Do you see that? Do you guys see this? I don't want you just sitting here just to clock in time. Is it making sense? I see some people nodding no. If it doesn't make sense, say something. Ask a question. How do I know something? What is going on? You guys feel free to ask questions. So we're talking about a phenotype. So we know that homozygous dominant is 50-50 and heterozygous is 50-50, but it's 100% round. So half round, half wrinkled? No, right? All heterozygous? No. All round? Yeah. They're all round for sure. Half heterozygous, half homozygous dominant. Well, for sure that's the best answer, right? Right? So you could have this could have, but you know, why couldn't the answer be D? Isn't D true? D's true. But what does it say? What does it say up here? So see, that's the trick. A lot of kids chose D, right? And they chose D because it's true. Yeah, it is true. And that's what they do to you on these tests. They give you right answers, but to a different question. So you have to make sure you understand the questions. Problem solving 101. The first step to true good problem solving. What are they asking me? What do I know? And then how do I solve it? Right? You have to have a plan. So what are they asking you? They're asking for genotype. You know this is correct, but it's not the genotype, it's the phenotype. This tells you the genotype. And it's the right answer. So that's the best answer. It's A. 6. The fundamental Mendelian process, which involves the separation of contrasting genetic elements at the same locus, would be called fancy language, right? But you know the answer. So the fundamental Mendelian process, which involves the separation of contrasting elements, different versions of the same gene, what would that be? Different versions of the contrasting genetic elements are what? Alleles. So we're talking about big A or little a, right? Isn't that what we're talking about? So we're talking about separating alleles. Separating what? Where are alleles? Where are these contrasting elements? Where do we find them? On chromosomes. So one is big little a, and the other is big A, and they're found on chromosomes. So the fundamental Mendelian process, which involves the separation of alleles, is called what? Segregation. Separating the alleles. And when does that happen? What have you learned that happens? When do we separate the alleles for... Well, go ahead. When does that happen? What process? In mitosis, you're right, we do separate alleles. But we separate them evenly, right? But when do we get independent assortment, which means we're not going to separate them together evenly? When do we get independent assortment? Not mitosis, but what? What process? It's not mitosis. So you're right, mitosis does separate the alleles, but it doesn't evenly, so each cell gets the same alleles. When do you mix them up? When do you not get the same alleles? When does it happen independently, randomly? Meiosis, good. So meiosis is when that happens. So when Mendel was talking about segregation of alleles, when he was talking about independent assortment, he was talking about meiosis. He didn't know that. They didn't know what the process of meiosis and mitosis were. But that's what he was talking about. Probabilities are calculated using the addition rule when they are mutually exclusive. A lot of people put are equally likely. That doesn't make sense to me. I don't know what that means. When they're independent, you use the product rule. So be clear on that. When two things like flipping a coin, if I flip a coin now, heads or tails, what are the odds of getting ahead? 50-50. Then I flip it again, and what are the odds of getting ahead? Heads? 50-50. And now the question is, what are the odds of getting the two of those together? Is one dependent on the other? If I get ahead, is it more or less likely that I'll get ahead the next time? Not really. So what I do is I multiply the odds of each independent event. So that's independent. So I know it's not the most exciting stuff, but you got to get this basic statistics in your mind. That is the product rule. So probabilities are calculated using multiplication rule when they are the product rule. Well, before we said the sum rule, but now what? When they're what? Independent. All right. So again, if it's independent, you multiply. If it's one or the other, you're adding. If you can only have one, if you have 4 or 5 things that can happen, but only one of them can happen, then you're adding the odds of each event. Does that make sense? You got to get that straight in your head because you're going to see this again. All right. What ratio typically results from the crosses dealing with a single genetic locus? So we're talking about a single genetic. I kind of listed it out for you here. I drew them out. So what are the odds of getting from a single genetic locus, which means what? Dihybrid or monohybrid? Monohybrid. So you're only talking like an A, little big A, little B, big A, little A, crossed with big A, you know, that kind of thing. So the different ratios, and we're talking phenotypic ratios, right? Dominant, recessive, you can have a 3-to-1 ratio, right? Phenotypic ratio. That comes from this. You see it, right? Does that make sense? 3 dominant and 1 recessive, so you have a 3-to-1 ratio. Or that same cross could lead you to a 1 to 2-to-1 ratio, right? Genotypic ratio. Did it tell you genotypic or phenotypic? In the problem, ask what kind of ratios could be associated with this. So you can get a 3-to-1 if it's phenotypic ratio. You can get a 1 to 2-to-1 if it's genotypic ratio. If you cross the heterozygous with a homozygous, you can get a 1-to-1, right? So 1 for the phenotypic and also 1-to-1 for genotypic, don't you? So you end up with 2 of the recessive trait and 2 of the dominant trait. You also end up with 2 heterozygotes and 2 homozygotes. So you get a 1-to-1 ratio. So these are the 3 main types of ratios that you can get when you're dealing with a single genetic locus. 3-to-1, 1-to-1, 1 to 2-to-1. First of all, I want to say if they're talking about a single genetic locus, you're not going to get 4 numbers. Do you see that? So 4 numbers automatically tells you it's a dihybrid cross. This is kind of ridiculous, don't you think? 12 to 3-to-1, 9 to 7. What they did here is they took these numbers and they put them into a monohybrid cross ratio to see if they could confuse you. They tried to confuse you in the answer choices. Did you see that? They made them close. They predicted your wrong answers. So you have to be careful because that's what they're going to do to you on the course exams. You have to read it carefully and answer the question you're being asked. All right. Are there 3 different genotypes resulting from a monohybrid cross? Or there are 3 different genotypes resulting from a monohybrid cross. How many different genotypes would there be resulting in dihybrid cross? So, right. So if you get 3 different genotypes resulting in monohybrid cross, what that tells you is they're heterozygote, right? 2 heterozygotes. How do you get 3 different genotypes? The only way is that both parents are what? Heterozygote. So if you're getting 3, you're going to assume then that the parents here are going to be heterozygotes, and so it's just a heterozygote cross, and this is where a lot of people messed up. So let's go ahead and go through this in detail. The people that didn't do well on the dihybrid crosses didn't know how to get this box. They didn't know how did you get the gametes? We did it in class. You did it for homework, but let's go ahead and do this right here and now and make sure you know how to do it. So if you're taking 2 heterozygote on 2 different genes and you're crossing them, just use the FOIL method that you learned in math to come up with the possible gametes. So you can get a big H with a big H, right? I'm sorry, that's not right. I'm messing up.
Who taught you that?
Huh?
Who taught you that?
Who taught me what? That was in math. You didn't learn math? You didn't learn in math? So you have to ask yourself, how many gametes can this creature make? So you take H and you combine it with a big A. So you have one of its gametes can be big H, big A. That's one possible gamete. The next is you do the outer, so that's big H with little a, big H with little a. And now you have to do the second. You see, we just did the first one. Now we're going to go do the second one. Little h with big A, right? And the last possibility is what? Little h, little a. So those are your 4 gametes. That's it. Now both of these parents can make the same gametes. Parent 1 and parent 2 can make the same gametes. You're lucky because then all you got to do is this. Big H, big A, big H, little a, little h, little big A, and little h, little a. And those are your 4 on top. And the 4 on the side are the same. Big H, big A, big H, little a, big H. Oh, that's not big H anything. Little h, big A, and little h, little a. And those are what you get across. For a total of 4 by 4, when you have a dihybrid cross, you have 4 by 4. And then you just add them together. Big H, big H, big A, big A. And you fill them all in every direction. Does that make sense?
Donuts are good.
Yeah. You guys go ahead and grab your donuts real quick. I'll pause the video. So now you got your donuts and some water. Let's take a look at this. So that's how you do it. That's how I got this. I got the dihybrid cross this way. And it says 3, and that's, how did I know they were heterozygotes? Because it told me I had 3 different genotypes, resulting 3 different genotypes, which tells me they're 2 heterozygotes, which tells me that the only way I can get 3 different genotypes, 1, 2, and 3, is if their both parents were heterozygous. So the question is, how many different genotypes would there be resulting in a dihybrid cross? I counted them. I just did it. I did the cross, and then I counted. How many different genotypes do I have? I have 1, big H, big H, big A, big A. I got the second one I counted was little h, little h, little a, little a. The third one I counted was big H, big H, big A, little a. Now, there were 2 of those. But I didn't count each one, right? Right? I only counted 1. This is different. These 2 are different from the rest. So this is one of the different ones. Huh?
So some of them are the same, so you don't count just.
That's right. Some of them were the same, so I didn't count them. What some of you did, some of you chose 16 because you were like, look, there's 16 different ones. But they're not different. These 2 are the same. These 2 are the same. Or this one's by itself.
Oh, okay.
This one's these 4 are the same. You see,
The same combination.
they have the same combination. So there's only 9 different There's only 9 different genotypes. We understand this,
You didn't finish that.
guys. Some of you are kind of what?
I need somebody to open some windows.
Open some windows. I think one more window if you wouldn't mind. No more than that. So people are going to start getting cold. I agree. Okay, chill. Literally. All right. Now, what is this? What would you? Someone asked a question
I
about.
said we didn't finish that. We started it, and then you did all of that, and then you took it off when you got done with
What are you talking about?
the dihybrid cross in
This
the corner.
one?
No, in the corner, all the way off the page.
This one?
No, on the other side.
Oh, I didn't finish this. I didn't finish this because I finished it here already.
Okay.
Why? You know, all you do is, as I said, let me review that. That's a good point. Let me make sure you understand what I'm saying. After you get your 4 gametes, they go across here and here, right?
Yeah.
Now, the only reason that the same gametes go across here, as you see here, is because they're both heterozygotes. So they're going to make both of them are going to make the same gametes, right?
Yeah.
Well, what are we talking about gametes? Remember, meiosis, right? Independent assortment, right? All right, so you got your 4 gametes, 4 on top, 4 on the side. Then you start to input what you got here. Remember, what are we talking about? This is sex. This is when you're taking the gametes from mom and the gametes from dad, and you're making a new kid. The new kid is inside each of these boxes. Different combinations, right? Who knows? Different outcomes. So these 2 are going to come together to form one And notice, what do we know about every living diploid creature? We know that you're going to have 2 copies of each gene. So here we only have 1 copy of each gene because they're gametes. But here this is a full-fledged creature, so you have 2 copies of each gene. So big H, big H, big A, big A. So you asked the question, why didn't we start with the A's? Why did we start with the H's? Because it's easier to keep track of them. So if I say H, big A, I don't want to repeat that. So I can write H, big A, and now I can make sure I do H, little a. So you see I'm doing the H's first. Could I have started with the A's? Could I put the A's on this side, H's on this side, started with the A's? I could have. That's just the way I wrote it. But what's key here is that you want to make sure you keep it. Look, a lot of you messed this up. You got yourselves confused. You did what I almost did at the beginning of this. You took the H, you went over here to the H, you put 2 H's here, 2 A's here. You know what I'm saying? That's totally wrong. It's this creature's making the gametes and this creature's making the gametes, and you have the gametes here, the gametes here. And then they combine. And you have to have, each gamete has to have 1 copy of each gene. Because then the zygote, the embryo, has to have 2 copies of each gene. Right? So if you have A, big A, big A, big A, big A, then all you do is have big A, big A, big A, big A, 4 copies of 1 gene, and that's not okay. And you lost the whole gene on top of it. So you have to follow this procedure every time, or you're just going to get lost. I mean, it's easy to do. I get lost sometimes trying to do it if I'm not careful. So you gotta keep that straight. So you're making the result is this chart. And the answer, of course, to this is 9 because there's 9 different ones. There are 16 different possible combinations, but some of these repeat. Look at number 6. Each of these is, all of these are big H, big A, little a, all the way down across the chart. Okay? Do you see that? I know it's a little blurry, but these 4 are the same. These 2 are the same. These 2 are the same. Etc. I think these 2 are the same. All right. So, and these 2 are the same. These 2 are the same. All right. So there's a lot of repeats. That's why there's only 9 different ones. Huh?
You put the little number, the same numbers are the ones that.
Yeah, that's what I did. That's how I kept it straight in my head. See, I'm doing it. I don't want to confuse myself, especially since I'm the teacher, because then I'll tell you something wrong and I'll be embarrassed and I'll have to correct myself. Hey, baby.
Sorry, honey, can I have the keys to the
Yeah.
restroom? Sorry.
Here you go, baby. Thanks.
I said I'm gonna start using shapes like triangles.
Yeah, you can use triangles. In class, I used triangles, like a triangle, a circle. You know, you can do that. But I just do I did that on the board. But what I did here is I just used numbers to associate them. All right, so let's go on to the next one. This one some people got wrong, and I really didn't understand why you got this one wrong. If you got this one wrong, I just don't get it, because this is really, like, it was really straightforward. A goes with T, man. That's it. A goes with T. So that's this one. A lot of people thought they got it right because it looks really easy, but they didn't pay attention to detail. So here, the detail got you. This one would have been easy. I think almost everybody got this one wrong. And it's not because you didn't know it. It's because you didn't pay attention to detail. Even the people I literally had 4 or 5 people get everything else right. The hardest stuff there is, they got it right. They even got the extra credit one wrong, right? And then they got this one wrong. And the reason they got it wrong is because it's really easy, and they did one little twist. So look, they have a 5' and they have a 3'. Then they tell you which of these match. Now I told you they're going to predict your wrong answers, didn't I? Yeah. I said they're going to predict your wrong answers. So the very first thing they did is they put AGCTAG, which is the matching sequence to this, right?
Oh, you supposed to start with AG?
Well, hold on. I'll show you in a minute what the trick was. They put that here. They put that here as number 1. You see it?
Yeah.
And so and then you're just like, Well, I thought that was it. It matches, doesn't it? Isn't that the code? That is the code, you're right. But 5 prime doesn't match up with a 5 prime. It doesn't. You see, it has to be opposite. It has to be 3 prime to 5 prime. So you have to switch it. So that on the 3 prime end, what you have to have is an A, and then the G, then the C, then the T, then the A, then the G, then you have the 5 prime. It's anti-parallel. This wasn't testing whether you knew that A goes with G and T and C. This was testing whether you understood the idea of anti-parallel. Do you understand that 5 prime matches with 3 prime and 3 prime with 5? It's an old trick. I got this trick when I was in college. I got this trick when I was in high school. This is a trick they throw at you all the time on ACTs, on standardized exams. This is something you got to keep an eye on. Do the prime ends match?
I remember it now. Because when we did
Yeah.
the lab,
Yeah, that's right. Yeah. Huh? That's just the way chemically they match up. They don't match up unless in any other way. If the 3 prime tries to match up with the 3 prime, they repel each other, so they don't stick. The hydrogen bonding doesn't work. Does that make sense?
make coffee. I'll make fun.
Man, there's nothing like good coffee. All right. Let's take a look at this one. Starting with a cross of these two, now we have 2 homozygotes, right? What proportion of heterozygotes would you get in the... And this got a lot of people. A lot of people got this one wrong. They predicted your wrong answer, and you got it wrong because you didn't see this one key phrase, and they did this several times to you. It's F2. They're asking for the F2. The F1 is the first cross. What do you get in the first cross? All heterozygotes. And you see they predicted it? You see how they predicted your wrong answer? So they were testing here whether you understood the first test they gave you is, did you understand what the difference between F1 and F2? If you didn't, you chose E. A lot of people chose E. Because you didn't notice or you didn't think about the F2. The F2 is when you take these two and cross them with each other. So you take 2 heterozygotes, big A, little a, and you cross with a big A, little a, and then you get some combination. And there it is, right? This is your F2. These are your F2s.
Okay.
So the question is, what proportion of heterozygotes do you get? You get half, right? You get 2 heterozygotes out of 4, 2 out of 4, or 1/2, 50%.
You got something.
Make sure you put the time. Do we all understand each other? All right. So don't mess that up next time. Look for F1. Look for F2. Make sure you understand how do you get the F2 if they give you the F1. You cross them, and their children are the F2. Are we clear? All right. Well, I'm sorry. Their children. You cross the parents, and their children are the F1, and you cross the F1s, you get the F2. All right. So just to be clear. It's the grandchildren is what you're talking about with the F2 from the original parents. All right. Genotype is to DNA as phenotype. Now, this is kind of difficult, and honestly, it can go in a lot of different ways. But the way I looked at it is genotype is a kind, right? Type means kind, and DNA is a biomolecule. So then they ask you, phenotype is a kind. So what is the biomolecule associated with the phenotype? It's proteins, right? Especially now, everybody understands that because that's how you get your traits. It's your proteins. Are we clear? That's the functional unit. Is this good? Yeah. Okay, so we understand. That was a tricky one. Honestly, if you got that one wrong, I wouldn't sweat it too much. Because, but the other point to mention here is they love to do this on the SAT. A tree is to grass as a human is to a door is to a building as a window is to something, I don't know, right? So anyways, they love to do that kind of stuff, and you have to think, what kind of analogy are they going for? All right, so think about that. But this was a difficult one here. just because it was really abstract. There wasn't a lot of rules you had to apply. So albinism is a lack of pigmentation in humans. We talked about it in class a lot. That's why one of the reasons I talked about it a lot is because I knew it was going to be on your test. Results in autosomal recessive gene designated, right? Little a. So recessive is going to be little a. Two parents with normal pigmentation. What does that tell you if they have normal pigmentation and they have an albino child? What does that tell you automatically? They have the trait. So it's big A, little a. So they're heterozygotes. See, this is what you're going to need to apply at the end in the pedigrees. A lot of thinking goes into this. This genetics is the most thinking you're going to get. It's very puzzle based. You have to think, and then every little step, any one of the little pieces that we're talking about is going to just ruin your whole answer. So the next question is, what's the probability of their next child being albino? Now you got to think. You have to figure out they were heterozygote. So there's a one quarter chance of getting an albino child. So they want to know what's the probability that the next child will be albino if they got one child that's the first is albino. So it's a 1/4, right? What are the chances of getting a girl? Is 1/2, right? Because either boy or girl.
Right.
It's 1/4 chance of getting albino.
Right.
So what's the probability the next child will be an albino girl? 1/4, 1/8, right? What's the probability that the next child will be? What's the probability that their next child will be albino? It's 1/4. What's the probability that the next child will be albino girl? 1/8. We got it?
Oh, see, I got the first part right, but I got the last part wrong.
Yeah. We gotta multiply the two. Because they're independent, right? Getting the albino gene has nothing. It doesn't affect if you get a girl. It's not sex linked or anything. All right. So now we're done. We're on second page, page 2. Now we're getting a little more into the rules, starting with the parental generation with the following genotypes. So now you have 2 of these genotypes based on the classic Mendelian inheritance. What's the expected phenotypic ratio among the F2 parents? So this would produce 100% heterozygote. Do you see that?
Yeah.
These are 2 homozygotes. They're mating. The F1 would be completely heterozygous. But look at what they're asking. They did it to you again. They're asking for the F2. They give you the parents. You had to figure out the F1, which were heterozygote, and this is it. So that's the second step. And you cross these two, and you get your F2s. What would the phenotypic ratio be for a dihybrid cross between 2 heterozygote parents? It's always 9 to 3 to 3 to 1. Always 9 to 3 to 3 to 1. It's one of the ratios I told you had to memorize. So this is always 9 to 3 to 3 to 1. By the way, you'll see this question later, but the only way to get 1 to 1 to 1 to 1 in a dihybrid cross is to have a test cross. F2, F1.
No, progeny.
Oh, progeny. Yeah, progeny.
So that's the boxes that's together.
Right. So that's a good question. So so let's go back for a second. Oh, I don't know how to do this. If I go back, it's going to try to get all confused. But when you're on the previous question when we did this, right? Where we did this chart, you had 16 blocks. Each one of these is a progeny. Each one is a progeny. So each one of these would be an F1 if it's the parents being crossed. Each one of these would be F2 if you're crossing 2 F1s.
So is F1 always heterozygous?
That's a great question. I hope there's I see some of you are, remember you don't have to be here, but this is really to get, did you sign out there? Okay. So you only get a heterozygous F1 is if your parents are homozygous. Okay, it's the only way you get heterozygous. So one parent's homozygous dominant, do you see that? and the other parent's homozygous recessive. So that's the only way you're going to get all the children being heterozygote. That's how you get 100% heterozygote.
So it's basically kids, question two, kids.
Yeah. F1 means kids. Actually, what it really means is filial. I can never pronounce it correctly, but
Spell it.
filial. It's Latin for brothers.
So F2 is like grandkid?
Yes,
Yeah.
of the original pros, right? All right, so let's look at 17. In the previous question, A equals plain fur, all right, and B equals long ears, and little b equals short ears, right? Are we good? Are we good with that?
Yeah.
So we know they did you a favor and automatically told you what the dominant recessive ones were. The dominant traits would be in the highest proportions, 93 to 3 to 1. So 9 plain long-eared in the F2 generation. So they said, what phenotypic class would be observed in the highest proportion in the F2 generation if you did in the F2 generation? So what's the F2 generation? 2 heterozygotes, the grandkids, the 2 heterozygotes getting crossed. So you get what proportions would you expect? What ratio would you expect? 9 to 3 to 3 to 1. And what would be the biggest? What would be the 9? What would be the 9? 9 what?
I never understood.
You didn't understand it?
No, I can't make the connection.
Yeah,
I understand the ratio, and then I understand that I have to cross, but I can't never make the connection. Put it together. The ratio.
Homozygotes.
All right. So we did it in class, but let me go ahead
Oh,
and
yeah,
do it
what's in
here.
that packet that we had over the break? They're talking about the 9 and 3 and 31, remember?
Yeah.
We talk about making the connection. I can understand both sides, so it's not good.
Well, let me say this. Let's do this. Like that, and then you have
That was a bad process.
this. Yeah, that's it. That's why I didn't want to do it now because we don't have that much time. But it seems like we have a lot of time, but we don't. And we've done this several times, but
Maybe if you start it, if you.
doing a dihybrid cross because one of the kids is saying that they understand this part and they understand the other part, but they don't understand how it goes together. So I'm going to try to... So you...
Wait, are you talking about when they when you did the little matching and you put the numbers in the circle? You talking about
Yeah.
That's the 9 and 85.
Yeah, that's the 9 to 3 to 3 1. What we're talking about, and I didn't want to go back and look at it, but what we're talking about, if we do it, I'm going to go ahead and do it, I guess. It might help somebody else, some of the people that are talking right now instead of focused, and it might help them, I don't know. So with the 9 to 3 to 3 to 1, you're talking about your... let me undo that and try to write with a thinner line. So when you're talking about making these, you should try to make it on your own right now and see if you understand it. This is the only way you're going to ever figure these things out is if you do it.
I got one question about this.
Yeah, go ahead.
Why do you like put the 2 A's at first instead of putting A, B, and
Yeah,
I used to do mine like that.
I
Then I
didn't
realized.
I do it this way, and this is the way, one, I was taught to do it this way, and secondly, it works better when you're trying to figure out what kind of traits you have. Because what you need to see when you're looking at to see what kind of traits you have is you need to see, do you have a homozygous or do you have a heterozygous, and how many, so it's difficult to see that if you don't have the same letters next to each other. So by putting the same letters next to each other, I'm able to much more clearly see where I have the recessive trait and where I have
Oh yeah, because it's separated.
it's almost like it's better.
you see, by putting them together, I know, for instance, that this has a homozygous recessive B, trait, whatever that is. All right, so let's take a look at this a little bit and then connect it to the problem. So this is what we should have gotten here. This is the heterozygotes, right? For this cross, the F2, the F1's being crossed, you get the F2s. And then they ask, the ratio is 9 to 3 to 3 to 1 for the phenotype. So what does that mean? Let's go ahead and let's do it. So what are the possible phenotypes you can have? You can have the big A blank, big B blank, right? So these are one phenotype. It doesn't matter what they get in the second space because you're going to get the dominant allele showing up. You can have the little a, big B blank phenotype. You can have the big A blank little b phenotype, whatever that is, and then finally you have the little a little b phenotype. So there's 4 possible phenotypes, right? That's why you have 4 possible phenotypes here. Are you making a connection now? These are why they got the 4 phenotypes there. That's why it's 9 to 3 to 3 to 1.
Okay.
All right, but let's go ahead and do it and make sure you understand and make sure you get why. So you got these 4. Let's count how many we have. Can we do that?
Yeah.
Let me move this over here and do it like this and get this big. All right. Well, let's just do it. Let's do it. What are you confused
I'm
with?
not confused about this.
Okay. Okay. So let's do this. Big B, big B. You know what? I should make this a little smaller so I count it. All right. So that's this phenotype here, right? So I can put a check, right? So I can say, okay, check. That's 1. You know what? Let me not put checks. Let me put slashes. So that's 1 of these, right? So that gets rid of this. Big A, big B, little b. Doesn't that go over here as well? So I can cross that one out. Big A, big B. Also here, it's the both dominant. So it's the dominant alleles are showing up so far. And this last one's also here. Do you get that? Are we okay with that?
Yeah.
All right, what's the problem?
So you took big A and big B and little A and little B and put it together?
No, what I'm saying is that all 4 of these are going to show up. The dominant trait shows up. There's a big A and a big B for all 4 of these. Do you not see that?
The last one was down.
Yeah, look. Watch. Big A, big B. You got a big A and a big B.
Oh, you did that from the side. I thought you was bringing the one from the bottom.
No, a big A and big B. They're coming over here. So this whole row actually is going to have all the dominant traits, right?
Yeah.
And so actually is this column, isn't it?
Yeah, I was about to say going down.
So this whole column also. So that's what? That's another big A, big B, big A, big B, big A, big B. So 3 more, right? 5, 6. So then where else do we see a big A, big B?
The third over from the
Right here, right?
Yeah.
So that's another one. 5, 6, 7. I got 7 so far. Do you see any more big A, big B's?
Yeah,
There's
yeah.
8, big A, big B. Any others?
No.
Big A, big B.
No. It's big a little bit.
It needs to be 5, 6, 7, 6, 7, 8. I'm doing something wrong here. Hold on.
Let me count mine. I did mine.
Where is it? Shh. 4, 5, 6, 7. Yeah, there's 9. I miscounted. That's all. So there's 9. So I missed the slash. No problem. So we got 9. Yes? Guys, this is where a lot of you have issues, so pay attention. If you just came in, which you shouldn't have, because I should have locked the door already, please make sure you sign in. Gosh, this is why I don't like to let people in late on Saturday school. So you got these 9. You see the 9? Yeah, that's where we get this 9. The 9 from 9 to 3 to 3. So the 9 of these. Okay. Now how many little a, big B something do we get? We would expect 3. Let's find them. Let me prove to you that it's 3. Right? Little a, big B something. That's here. That's 1. Here's another one, and there's a third one, isn't there?
Yeah.
So 1, 2, 3. That's your third. Now, big A, blank, little b, little b.
1 or 2. No.
We expect 3. That's what we expect. Somebody's saying it. That's exactly right. Big A, blank, big B, little b, little b. There's one there. There's another one here. And there's a third one here. You see it?
Yeah.
So 1, 2, 3. So that's 3. And then the last one is this one, which is little a. That's the only homozygous recessive you got. So that's why every time you now pay attention. That's why you always get 9 to 3 to 3 to 1 when you do a dihybrid cross with heterozygotes.
Because it's only one of those.
This is only one of these, and then you always get this combination. And I don't care what the trait is. I could be talking about horns or little a color of skin or height or anything. The trait doesn't matter. I could copy this and apply it to every problem. The letters can change, A and B and C and D or F and G. It doesn't matter. The ratio is always going to be 9 to 3 to 3 to 1. 9 dominant traits, 9 of the dominant traits, 3 of the dominant traits on one and recessives on the other, 3 of the dominant traits of the other, recessive on the first, and only 1 recessive on both.
So that's what you do in a dihybrid cross with heterozygote.
Very good. That's when you have a dihybrid cross and they're both heterozygote. You're always going to get 9 to 3 to 3 to 1. If you knew that, you would have got 6 answers. Probably, if you understood this, you would have got 6 answers quickly on the test correctly. Where'd she come from?
Oh, I did just see her.
I just like, what happened? I didn't even. All right, so let's take a look at this. Let's take a look at this. And so, remember to sign out if you're leaving. Make sure you signed in if you came in. Miss Perez wants to make sure you know that we know where you are, and your parents are going to want to know as well. All right, let's take a look at this. So in the previous problem, remember there's that 9 to 3 to 3 to 1 ratio, and that's why I said the dominant traits would be in the highest proportions. So you have 9 plain long-eared in the F2 generation. Are we good?
Yes.
All right, so that's why this is plain long-eared. How did I know it's plain long-eared? Because the dominant's plain and long is plain, so it's plain long-eared. So are we clear?
Yes.
All right, so let's move on. You see how you could have, if you would have learned these ratios, like I asked you to, if you would have understood, guys, if you didn't understand next time, you don't understand, come and see me before the test. That should have been, now I have to admit, this is a tough test. I told you that, right?
Yes.
But they're not going to take it easy on you on that end of course exam. Now, let's take a look at this. A species of mice can have gray or black fur and long or short tails. So gray or black, long or short. So that means that these are 2 alleles, right? Now here's 2 alleles. Do we understand that? The cross between a black-furred and long-tailed mouse and gray-furred short-tailed mouse produced black-furred long-tailed offspring. What does that tell you?
Yeah, homozygous recessive.
They're telling you big G is black, little g is gray. Big S is long, little s is short, meaning what? Big G is dominant, black is dominant, and long is dominant. So what does it tell you if you get black-furred, long-tailed, and you produce all black-furred, long-tailed offspring? You're taking black-furred, long-tailed mice and gray-furred, short-tailed mice. So what are you doing? You're crossing 2 homozygotes. And then you're getting all 100% heterozygotes. Right?
Yes.
That's what you should see when you read that. So using the G symbols for long and short, what would be the genotype of the gray-furred short-tailed mouse? That one's easy. That's this one. Little g, little s, little s. Done. That problem was easy. 19 says, referring to question 18, how many different gametes will the will the black-furred long-tailed mouse produce? All right. Black-tailed black-furred long-tailed. That means in P1, the parental generation. How many different gametes will it produce? It's only going to produce one because it is dominant. It's homozygous dominant. There's only one kind of gamete it can produce. A big G and big S. That's all it can make. It'll make 4 of them, but they're all going to be big G, big S. Are we clear?
Yeah.
All right, so there's only one possible type of gamete. They didn't say number. Remember, they always produce 4 gametes. Now, number 20 says, referring to question 18 again, how many different gametes do the black-furred, long-tailed F1 now? The F1 are heterozygotes. So they're going to produce 4 different ones. And how did I find these? I just went through it with you twice, didn't I? So you should be able to find those 4 quickly. So there'll be 4 different ones. So what would the dimensions of a Punnett square depicting dihybrid cross be? 4 by 4. We've done it, and here it is. I did it again. I did it for you here, but we've done it now 4 times just this morning. So it's a 4, 1, 2, 3, 4 by 1, 2, 3, 4. Why? Because it's a dihybrid cross, right? And you have to assume they're heterozygotes. You have to remember dihybrid crosses don't always have to produce 4, a 4 by 4. Because look at the next question. What would be the minimum? The minimum. And this problem actually was a weak problem, I think, because they didn't specify heterozygote. So it could have been this. You could have done a 4 by 1. You see. And they actually predicted that answer, didn't they? They predicted that ratio, that answer right here in A. Some people did choose A, and I actually had half a mind to give it to them right. But it's not the best answer. 4 by 4 is the best answer, so I went with that. So what's the minimum dimensions of a test cross with a dihybrid parent? Now that's a little better because they're telling you it's dihybrid. It's a test cross tells you what? Homozygous. Homozygous recessive. So you have something being crossed with homozygous recessive. So the homozygous recessive can only produce one type of gamete. That's little g, little s. And there it is. Where the heterozygotes can produce 4. So there's your 4. So I kept the letters the same for your ease, and you can see that the minimum dimensions will be 4 by 1. And there it is. Now to determine if an organism with a dominant phenotype is heterozygous, what do? You cross it with a homozygous recessive. You do a test cross. All right, and if you get 4 different, or you get, if you even get one of the recessives, in this case here, right, then you know it's heterozygous. But if you get 100% the dominant, then you're saying it's probably homozygous dominant. Okay, so let's take a look at now the F1 dihybrid cross. So there's the dihybrid cross again, where big W is round, little w is wrinkled, big G is yellow, little g is green. What is the probability of obtaining individuals that are wrinkled and green, true breeding? What does that mean? Good. I'm glad somebody admitted it, because I know that a bunch of you don't know because you got the answer wrong. So what is the probability of obtaining an individual that is wrinkled and green, true breeding? What does that mean? No, it means it's not hetero, it's homo. Homozygous. So, and wrinkled and green is what? Recessive. You see that? So they're recessive. So that would mean little w, little g, little g. What's the probability of getting a homozygous recessive individual? See, I drew it here, right? So true breeding means homozygous. That's what true breeding means. It's the second time I think I mentioned today. So homozygous means true breeding. True breeding means homozygous. Make sure you get that. So here you have this little w, little g, homozygous recessive. What's the chance of getting that? If you remember that 9 to 3 to 3 to 1 ratio, you remember there's only one homozygous recessive for both traits. That's that bottom corner one, right? If you did your dihybrid cross array, you copy it down, or you see it on the answer sheet that I sent you earlier. You see that the one homozygous recessive is the bottom right hand corner. See, 1 out of 16. That's why it's 9 to 3 to 3 to 1. So it's 1 out of 16. And notice again, I want to emphasize again that they predicted wrong answers. They gave you 9 out of 16. They gave you 3 out of 16. So if you don't remember that homozygous recessive was the 1, you might have put 9 or 3, right? So if you didn't understand that, you would have gotten the wrong answer. Also, they put 1/4, which you know is from the monohybrid cross, and the 50%, which you generally only get from the monohybrid cross as well, right? So they're really kind of throwing in some. They're asking, does this kid really understand what's going on? Number 25. What genotype is present most often in the progeny F2 generation? So here's your cross. Again, heterozygotes, right? What is the one that you're going to see most often? It's going to be this one, right? What is the genotype present most often among the progeny of the F2 generation of a dihybrid cross? So do the dihybrid cross, and you'll see that what you're going to see most is big A, little a, big B, little b. All right, moving on. Barney and Betty are about to have her first child. They have normal feet, but Barney's father had flat feet, and Betty's has a child from a previous marriage who had flat feet. What is the probability that they will have a boy with flat feet? Well, you know what would have helped? Is if you did a pedigree first. Make sure you sign in if you're just coming in, please. I shouldn't let him in. I don't know why I didn't lock the door, but make sure you sign in. So there's a sign in somewhere. Sign in. Make sure you put time in, and if you leave, time out. We only have half an hour left to finish this genetics, so. All right, 26, number 26, if we're looking at it, the best thing to do is to start with a pedigree. It helps a lot. So Barney and Betty, male, female, they're getting married, they're having kids. Betty, they're trying to have their first child, so they're looking at this. We don't know if it's going to be male or female. They both have normal feet, so and the father has flat feet, and Betty, we don't even know if it's dominant recessive yet, but Barney's father has flat feet. So now let's go ahead and take a look at Barney's dad. He had flat feet, right? And we're worried. This is Barney, and then this is Betty, okay? So Barney's dad had flat feet. Guys, what's going on? All right, can we? So Barney's dad had flat feet. Let's just give him the character. We don't know if it's dominant or recessive yet, right? Barney and Betty don't have it. Now Betty has a child from a previous marriage. So Betty was married before. And so let's go ahead and do that drawing. Rather than getting complicated, let's just do this again. Betty was married previously to somebody, and from a previous marriage who has, and that child, whatever it is, male or female, I'm just going to say female, has flat feet. But they didn't have it. The parents didn't have it. What does that tell you automatically? She's a carrier. So what is flat feet? Flat feet. Is it big F or little f? Little f. It has to be little f. Because you skipped a generation. It's recessive. So Betty must be big F, little f. Husband must have been big F, little f. The child must have been little f, little f. So Barney's dad must be little f, little f. His mom must have been big F, made blank, whatever that was, he was big F, little f for sure, right? And therefore, what is that? Betty, we know is big F, little f, right? And then her child, what are the chances of getting her child? Well, you got it. I used N. I used normal instead of F. But whatever, you'd have to do it here, right? This is it, right? 2 heterozygotes. You have 1/4 chance of getting flat feet. So doing the pedigree got you the answer, lets you figure out what was it recessive or was it dominant. And once you knew that they were heterozygotes, then that lets you figure out that they had a 1/4 chance of getting a child with flat feet, and they want to know. They actually want to know what's the chance of having a boy with flat feet. We know it's not sex linked, at least as far as we know.
Yeah.
So what's the chance of getting a boy with flat feet? You'd have to have 1/2 because it's a 1/2 chance of getting a boy, 1/4 chance of getting flat feet. So it's 1/8 is the answer. So you're multiplying because they're independent, right? It's just a lot of work for that problem, wasn't there? That's why you guys got 4 days on this test, because it was real tough. Yeah. Yeah, go ahead.
So how do I get the 20%?
You need to, I don't know. I don't want to talk about that now. I do know, I just don't want to talk about it right now. So, not when I'm recording and trying to answer the questions on the test. All right? It's not, I'm okay with that. You don't have to apologize. In the previous question, Barney and Betty learned that they will actually have twins, dizygotic twins. Dizygotic twins means they're fraternal, so they're not identical. What is the probability that they will have a boy and a girl, both of them flat-footed? So both of them having it. So the boy, having a boy-girl, there's 2 ways to do this. You can get a boy and a girl, or you can get a girl and a boy. So both the chances of getting a boy is half, a girl is half. So that's why you get these 2 halves here, right?
Yeah.
And then there's a 1/4 chance of getting flat feet. So what you do, you multiply these across. So that's 1/16, right? 2 times 2 is 4. 4 times 4 is 16. So it's 1/16. 1/16. That's 2/16, which is 1/8. So your chance of getting that is 1/8.
Wait,
It's that multiple. See the product rule and that sum rule?
oh, see, I just left that 116.
Yeah. Yeah. I mean, people have all day, so what the heck? Microbiologists who determined the DNA genetic material, but we said it was Oswald. Geneticists who discovered that genes could actually move, it was Barbara McClintock. And biochemists who discovered restriction of nucleases, Herbert Boyer. I gave you those answers. You should have got them all right. So it's because we didn't really hit that very hard. Okay, so now let's take a look at what produces. This is an easy one. What produces a 1:1:1:1 4 ones ratio? It's only a test cross. Take a look at it. Take a look at it. Your ratio is 1 of these to 1 of these to 1 of these. And notice that they're phenotypic ratios. They're all going to look different. You see that? This was going to have the dominant trait for the A, the recessive trait for the B. This has dominant traits for both. This has recessive for A, dominant for B. This has recessive for both. But you're only getting it's only a 4-to-1 cross, so you're only getting a 1:1:1:1. It's the only way you get this ratio is with a test cross. Only way. Test cross with a heterozygote. If you got a test cross with a homozygote, you're only going to get one type. You have 100% of the dominant. Does that make sense? Yes. So this is a test cross with heterozygote. It's the only way to get this ratio. In your own words, what is the meaning of a term allele? Anything that alludes to the fact that allele is a different version of a gene.
It was number 1. I just copied that.
All right. Yeah, and honestly, this answer, she's right. This answer was in question number 1's multiple choice. So all you have to do is copy the number 1 answer.
In your own
But
words.
if you got it right.
Yeah, if you got 1 wrong, well then it didn't help you. All right. Black and white collaboration cows. This was difficult because of how they wrote it. You would think black and white, there's two different traits, right? You're thinking codominance, right? Right? You're thinking codominance, like the pink, like the black, the red and white flowers, like all the other things. But this is not codominance. See, I'm glad you did, because it was tough, because it said black and white. I started this problem, I was thinking codominance. Then I reread it and I said, wait a minute, this is not codominance. So black and white coloration causes dominant. You see, this is the key right here. This is the key. That this is dominant to brown and white. So this coloration, black and white, is, let's say, big B, and brown and white is going to be another one, the recessive trait. Do we all see that?
Yeah.
So this is recessive, and this is dominant. Really, all I had to do is big B, little b, but, so I don't know what, I just put the little w because of the white, right? So I just thought it might be, I shouldn't have done it, but I did it, so whatever. It's just big B, little b. Big B is, so you could have wrote it any way you wanted. That's not the answer to the question. I just wrote it this way just to keep it straight in my mind. But you could just say big B is black and white and little b is brown and white. That's all you need to do. That little w does not need to be here. I don't know why I did it. I just did it. It kept it straight in my head somehow. But it's not necessary. All right? These are not necessary. If that confuses you on the answer, just get rid of it. So, what you got, now read the problem. It tells you after you cross these a lot, you get, pay attention because we're running out of time, 8 black and white and 3 brown to white. So you got 8 to 3. That's what you got. You got to remember that when we do Punnett squares, these are predictions. These are ratios. They're never 100% predictive. You could have a 1/4 chance, like in sickle cell, you could have a 1/4 chance of having a child with sickle cell disease. But you could have 10 kids that don't have it, or you could have 10 kids and they all have it. When we're talking about these crosses, this Punnett squares, we're talking about the odds. You know, people beat the odds all the time. And you know that there are other families that don't beat the odds. They do the opposite. They get this disease more often, right? So it's all about odds. It has nothing to do, and you want to call it luck, go ahead. I don't like the word luck, but that's fine. It's all, the way randomness works is that it's random. So we can't really tell. All right, what is the most probable genotype of each parent? Well, that's easy. You can't get the recessive, right? You can't get, because they say you get 3 that are recessive. How are you going to get the recessive if the black and white cows were 100% dominant? If they had big B, if they were big B, you could not have any children that were wait a minute, let me see. That's not true. What I'm trying to say is that black and white cows were crossed. So the 2 black and white cows, that means what? The black and white cows were what?
Dominant.
They were dominant, right? So because they're black and white, they're dominant. They have the dominant allele, so they both have the big B. There is no way they could have any brown and white children unless both of them were heterozygous. Does that make sense? Because they both have to have a little b to give in order to get a recessive progeny. Does that make sense? So that's why, and I explain it here, right? So what is the expected genotypic and phenotypic ratio expected for the progeny in the previous question? So you would expect to see what? Genotypically, and some people answered one, but not the other. Okay, some people gave the genotypic ratio, didn't give the phenotypic ratio. So the phenotypic ratio is what you see. You get 3 of these. 3 of these have the dominant trait, which is what? Black and white. So 3 black and white to what? 1 what? Brown and white. genotypic ratio, it's 1 homozygous to 2 heterozygous to 1 homozygous, 1 to 2 to 1. That's one of those ratios you had to memorize. Yeah, that's happened a lot. So just make sure you understand what's going on. You're going to see this on both your standardized exams that you're about to face in a couple months. So a certain type of congenital deafness in humans is caused by a rare autosomal dominant gene. This is important information. You should underline it. You should write it. You should make sure you realize that this is autosomal dominant. I saw people doing X-linked. I don't know why. Yeah. Autosomal means it's not X-linked. It's not sex-linked. So it's just your normal, everyday, big D, little d. Right? We're not dealing with the X gene, the X chromosome. In mating involved in a deaf man and a deaf woman, so they both have what? What do we know from deaf that they're both deaf? They both have a what? Big D. They don't know the other one, right? We don't know the other one. Could all the children have normal hearing? Well, we know that both parents are big D blank, right? And we know that little d means hearing. Now, if they were both heterozygote, there'd be a 1/4 chance of having a normal hearing baby. But is it possible that all their children, they have 100 kids, and all their kids are normal hearing? For sure, it's possible.
Yeah.
It's unlikely, but it's possible. The key word here is it possible? Could. Is it likely? Absolutely not. In fact, I said if they have 5 children, the odds are 1 in 1,024 that you would have all 5 children with normal hearing. But do we see it happen? Sure. But when you look at 100,000 births with people with this genotype, you're not going to see that. Yeah.
So both their parents can't hear, right?
Yeah.
So how is it even possible for them to pass down the genes that they don't have?
It's a good question, and the reason it's possible is because it's autosomal dominant. If they were heterozygous, see, if it was a recessive gene, it would be impossible. Because if it was recessive and they were deaf, then they would both have little d, and the only thing they could give is a little d, right? But because normal hearing is recessive in this case, they can carry the gene for normal hearing even though they're deaf. All right, so now the white-eye gene in Drosophila is recessive sex-linked. You see, this is autosomal. We just talked about it. The normal stuff we've been talking about all year, or for about 2 months now. But now this is the recessive sex-linked. So I know it is. We have 2 windows open. I guess, could you open up a third one, Austin?
Wow. I told you we should.
Only because you're complaining again. I'm doing it.
No, you said it's happening.
So assume a white-eyed female is mated with a wild-type male. Now, recessive, we just said R is recessive, and red is dominant. All right, is mated with a wild-type male. It says here, white-eyed gene Drosophila is recessive sex-linked. Assume a white-eyed female is mated to a wild-type male. Wild-type means it's not recessive. In this case, wild type just means what you see in the wild. What would be the phenotypes of the offspring? So now you have to add the added consideration that the boys and the girls are not going to have the same genotype. So if you have again it says white-eyed female, so the female has a recessive on her X's. You see that? She's white-eyed. Be careful. Oh my God! Don't, Daza, be careful. Daisy, sorry, Stacy. Just be careful. Come down carefully. Oh my.
You need some water?
I got it. Thank you. Children, this is why we're here on a Saturday, and there's so many of you. Let's try to focus, please. I know it's been a long day. It's 9:46, but you got to focus.
Oh, I'm not
All right. When we cross these two, the male is red-eyed, and the female's carrying the recessive white-eyed. So all their female children, 100% of their female children, are going to be red-eyed because dad's going to give them the dominant trait. Do you see that? The dominant gene. Does everybody see that? That all the females have the dominant gene. Pay attention, even if you have to leave in a few minutes, just pay attention first. Now, dad is going to give a Y to his boys, so he can't give them the dominant gene. So all his boys are going to have the recessive gene that mom carries. So all the females' male children are going to be white-eyed. And I miswrote here. You see, I wrote this incorrectly. This should not be female. This should be white-eyed what? Males. Make that correction, please. Make that correction.
Is it wrong to write the allele, then X and Y? Because I feel like the big R, then X is good.
I would say it's wrong because what's going to happen is that you're not going to realize, you're going to mess it up, that this is, the boys are white-eyed. The girls are red-eyed. And you're going to mess it up if you don't do it this way. So I'll say this, do it the way I'm showing you when it comes to this sex-linked stuff. But as long as you give me the right ratios, I'm not going to care. However, I would really, really consider using this notation. What would be the phenotypes of the reciprocal cross? That means, obvious reciprocal, I told you during the test, means opposite. So instead of white-eyed female, you have a red-eyed female. Instead of a red-eyed father, you have a white-eyed father. Well, you're going to get, when you do that, what do you see? You get 50% of the females are white-eyed, right? And we said if it's heterozygous, 50% of the females are going to be white-eyed, and 50% of the males will be white-eyed, right? So, but if it's homozygous red, then what we're dealing with is red. All the females have red eyes, but they're heterozygous, and all the males have red-eyed, and that's all they are. All right, so 25% of your progeny will be red-eyed female, 25% red-eyed male, 25% white-eyed female, 25% white-eyed male, and that's what you'd end up if it was heterozygous. Are we all good with that then?
Yeah.
Let's move on to this one. Give a reason why the relationship between genes and traits is often complex. Gosh, guys, I told you that we went through this in the chapter reading. I really was disappointed that there's so many people got this one right or wrong. This should have been pretty straightforward. One gene can influence many traits. We can have polygenic inheritance. We can have sex link. There's so many different ways of inheriting how genes and traits show up. How traits show up with the genes. Now here, this one was, the next question was actually pretty easy if you broke it down. They have a lot of information, but it pretty much broke down to saying, look, we got, in the end, we have 116 males have black. All the males are yellow. All the males are yellow. What does that tell you?
It's dominance.
No. No, what does it tell you?
Wait, what you just asked?
The first thing is, I wish you'd all listen, and God, I feel like some of you are wasting your time here. So we have 240 females are black, 116 males are black, but none of the females are yellow.
Oh, it's down a Y.
So yeah, I'm just going to keep going. So what we have then is 116 males that are black and 123 males that are yellow, but only the males are yellow. What does that tell you about yellow? It's sex-linked. It's X-linked. When more males have it than females, it's X-linked. That's it. And it's recessive, and the way you know it's recessive is they actually tell you homozygous wild type female, yellow-bodied males, all F1s, right? So they tell you it's homozygous wild type. So they're already telling you which one's dominant, which was that it's the yellow's recessive. But now, how is it inherited? It's inherited by an X-linked method, and we know that because more males have it than females. In fact, none of the females have it. Yeah.
Why is it not called Y link?
Okay, because it's not on the Y, it's on the X chromosome.
I thought X
X and Y male and XX female.
We talked about that. If you wanna come after school next week, I can go into that. But the X, look,
Oh, no.
no, I don't have time to get. We have one whole other one to do. I'm just gonna say this: the X chromosome, when we're talking about X-linked, that you have a difference because Dad gives a Y. If you have a Y chromosome, it's always if a Y-linked gene, which can happen. It means only males have it. So, okay, yeah, but if you read in this question, you asked it specifically in this question. Is that what you're saying? Yeah.
Why is it not called Y link? Why is it the X? That's what you're not getting what I'm saying.
Okay.
Since Y is on.
Say that again. What'd you say?
No, I'm talking.
In this particular problem, I would say X-linked recessive because it's more common. There's nothing really in the problem that tells you that it's Y. That's not Y-linked. So
Oh, but females can get it. That's what you're saying?
that, and what I'm saying is that more likely than not, it's a female. It's an X-linked trait, and it's kind of alluded to in the problem because they say homozygous. You see here, they say homozygous wild type female. So that suggests that there might be a heterozygous, right? So the problem suggests that it's X-linked. You're right that the Y-linked is a possibility given that we don't know where it's carried and you're only seeing males. So it could definitely be a Y-linked trait. You're right on that, okay? But when you're looking at the problem, it suggests that it's X-linked, and X-linked is more common. So that's why I went with that answer, okay? So I agree with you, but I just didn't want to talk about it right now because we got to get finished this. All right. Yeah, I know, we got a few minutes. I think we can finish it if you pay attention. I'm not going to do this because this is just, all you had to do here, let me quickly tell you this. All you had to do here, Abdul, all you had to do here was do the dihybrid cross and count. How many of these are yellow? All the big Y's. There it is. How many of these are little l, you know, how many of these are this, how many, you just count them and reduce them, and you get your, how many in each. So that's not really difficult. I'm moving on. This was not that difficult. This is actually the stuff that most kids get easily if you put some work into this. But a lot of you messed this up really badly. I mean, it was really one of those things that was like, oops, I can't believe that happened. So question number 1, real easy, and you have to do the following process. You got to be able to do a pedigree. Almost guaranteed in your Procore exam you're going to see one of these, and almost guaranteed you're going to see one in your end of course exam. So you got to be able to do this. So in this pedigree, the first question is, how is it inherited? Is it sex-linked? Is it recessive? Is it dominant? Well, I don't see it skipping a generation at all. If a person has it, the next generation has it. There's nowhere where you don't see it in the whole generation. So I'm going with dominant. I'm going with the most likely to say dominant. I don't see where the number of females and males have a large difference between males and females. I know that females have one more female has it than male, but it's not, guys, just because we're running, it's 9:56 and we're about to switch sessions, it doesn't mean we want to waste the last few minutes. So just listen to me. This is extremely important. I can't stand that I'm talking about this behavior on a Saturday.
What behavior?
The other kids.
All right. So when we're looking at this pedigree, I'm going to choose dominant, and then I'm going to say autosomal. Now, if it's dominant and autosomal, then let's see if it fits all our data. We know then that this person had it. That means it's big A, little a, and that because only now in this case, because I said it was dominant, if it's filled in, I have a big A. I saw people reverse this all day long every time I graded these. So if it's big A, if it's filled in, it's a big A, but only because I said it was dominant. How did I know it was dominant? It did not skip a generation. Every generation had it. Someone might say, well, what about these guys? These guys don't have it, but none of their children have it either.
Right.
So the gene did not skip a generation. Now, the other thing, so that means that every time they don't have it, it's recessive. So little a, so I don't need to explain that. Every time they have it, it's a big A for sure. How do I know if it has a little a? How do I know if it's heterozygous? How do I know number one is heterozygous? Because one of the kids don't have it. Here, this kid doesn't have it. So this kid's little a, little a. He got one little a from mom. Where'd he get the other little a?
From the daddy.
From dad. So dad has to have a little a. So he has to be heterozygous. And you do the same logic for all of these. I'm moving on to the next problem. So let's take a look at this. This says, and you see that they ask you, I should point out that they ask you, Roman numeral 1, number 1, that's Roman numeral 1, number 1, then Roman numeral 1, number 2, then Roman numeral 2, number 1, then Roman numeral 2, number 3, Roman numeral 2, number 3. So they ask you that. I filled in everything and I showed you the logic behind it. So read that, please. In this one, please take a look at this. Look at this carefully. These two parents don't have it. They have a child with it. So what does that tell you? That it's recessive. The parents are carriers, so it's a recessive trait. Do I see more boys than girls have it? No, so it's autosomal. And then the rest of the logic is the same as the first problem. But you have to answer those two questions first. Now I'm going to go over the last two problems in this. Before we go on to protein synthesis, I'll let you guys switch out sessions if you want. First thing we're going to do, now that the break is over, first thing, it's now 10:11. Did we sign in?
Yes.
All right, so make sure you signed in. Miss Perez wants this. So before we get on to protein synthesis and going over that exam, what I want to do is go over, finish this last essay on the genetics portion, if you don't mind.
What happens if I stayed here both times, like I didn't need to sign in again?
No, you just sign out once, right?
Yeah.
All right. So remember, to get the 20% boost on the genetics portion, all you have to do is rewrite the answers in your own words, right? All right, so this problem seemed like it was hard, but actually it was pretty easy. But it just took a lot of steps. Say that again?
Behind you there was a shark whale, not foul.
Oh, okay, thank you. That the iPad must have clicked out.
Shark, my bad.
Okay, we're recording now. Remember, this is going on YouTube, so don't say anything that you're going to be embarrassed about. Make sure you're signing in. Make sure you're signing out. All right, so the short essay, this is the last little bit we got to finish, and then we'll move on to protein synthesis. In the short essay, you'll see that this problem looks complicated, but it's really not, right? So they say they took a longhorn and they crossed it with another longhorn, and they got 39 longhorns and 2 shorthorns. Now, what does that tell you? What does that tell you? Not you. What does that tell you about whether this is dominant or recessive? Longhorn is probably dominant over shorthorn, right? Also, and why do we say that? Because these are both longhorn. So what is shorthorn? What's happening to shorthorn in this case?
One of the parents was heterozygous.
Well, both of them had to be heterozygous, right? And you skipped a generation because these were both longhorn, but it has a shorthorn parent. child. So that skipped a generation. So that means Shorthorn is recessive. You see how that works? It's basically a pedigree chart in a different form. So you have these two parents producing that are both Longhorn, producing a Shorthorn, at least one Shorthorn. So that means that these parents are heterozygous and that Shorthorn is recessive. So that's what this taught us, A. B is Longhorn and Shorthorn, so that we know now that Shorthorn is little s, right? Or I think I used little l. So little l, where the Longhorn, we don't know what it is. But what would you think it is based on the fact that when we cross these two, you get no Shorthorn?
That is the one that is homozygous.
Exactly. That we know now that because of this, we know that Longhorn is homozygous dominant, and we knew already that Shorthorn was homozygous recessive. So all of their children are what? If we take a cross, we do a quick Punnett square. All this is a Punnett square. Do it if you don't remember it. All you have to do is say, well, I have a Longhorn that I know is dominant. I know I have a Shorthorn that I know is recessive, and they're homozygous. I know that for a couple reasons. First of all, I get no Shorthorns when I know Shorthorn is recessive, so I know that it has to have it has to be homozygous. I know that I get no Shorthorns when I cross it. The only way to get 0 Shorthorns is if the dad or the mom, whatever it is homozygous dominant, which leaves us to 100% heterozygous. But of course, heterozygous are going to show the dominant trait. Okay, Shorthorn versus Shorthorn, and I get 40 Shorthorns and no Longhorn. That data supports my concept that the Shorthorn is recessive, right? Because when I cross Shorthorn with Shorthorn, I get no Longhorn. It wouldn't be possible because the Shorthorn were all little s. Please sign in if you're staying. So they ask us quickly, they say, do you see how easy that was, right? That was pretty easy. And I gave you this as extra credit. Not a lot of people tried it. I think they just got confused because of how the data was presented. Predict the results of F1 mating from cross B. So B would be what? It would be Longhorn and Shorthorn. The F1 mating would produce F2s, and we actually did this already, right? We did it right here. What would be the suggestion would all be, right? It would all be 100% hetero. All right, so that's it. That's the answer. They're all. I did it again. And then what the next one says: what are the possible explanations for the 2 Shorthorn jackalopes in cross A? Everybody understands jackalopes are made-up creatures, right? Yeah. Okay, because some people might think that they actually exist. They don't. I know it's funny, but you never know. What are the possible explanations of 2 Shorthorn jackalopes for cross A? We look at A. How is it we get these? Well, because they're heterozygous, and I explained it all here, and that should answer that question. All right, this one is a pedigree. Alice and Ralph, right? Decided to start a family. Yeah, you got a question?
Didn't it say an F1?
Yeah.
Wouldn't that be the show drop?
Yeah.
The F1,
Yeah, because those would be the P ones.
right, cross B. So that would be 2 heterozygotes. So if you think about it, Longhorn, Shorthorn, the F1 generation, that's this one, Longhorn, Shorthorn, produces what? And this is your F1. So cross 2 of these, you're crossing 2 heterozygotes. And so you get, this explains the 2 Shorthorns, right? Does that make sense? Okay. So Alice and Ralph have decided to start a family. Ralph has normal vision, but has a brother who has rare inherited form of blindness. Rare inherited form. And Ralph has normal vision. Neither Ralph's parents or his 2 sisters are blind. So what I like to do is I like to do a pedigree. I like to do a pedigree. Neither Ralph's parents nor his 2 sisters are blind. So where are we? So I said, there's Ralph. His two sisters are not blind. His parents are not blind. But then it says Alice. Alice has two brothers and two sisters with normal vision, and neither of her parents are affected. Right? Draw a pedigree to illustrate the situation where standard symbols in males affected and unaffected. I think maybe I misread this. Ralph has a normal vision, but has a brother who has a rare inherited form of blindness. There's Ralph. He has a brother who has blindness. Neither of his parents are blind. So what does that mean for this blindness? Yeah, it's recessive because it skipped a generation. So that means his parents have to be carriers. They have to be heterozygous. Yeah. No, there are dominant forms of blindness. So Alice has two brothers and two sisters with normal vision. So two brothers and two sisters. This is Alice, normal vision. And neither of her parents are affected. Draw a pedigree to illustrate this. Okay, so this is the pedigree. Do you see that? All we know is that she's normal. We don't know anything. Now, odds are that she's big N, that she doesn't have the recessive trait. That's the odds. The odds are that these two parents are big N, since none of their children are. But there's no guarantee of that. I say again that odds are random. So if you were forced to say, is Alice homozygous dominant or is she heterozygous? If you're saying those are your only two choices, then you'd have to say homozygous dominant, because that's the evidence that you have in front of you. But there is a possibility that she's a2carrier. It's just small and just didn't show up. So remember, you can have, just like in the sickle cell example, yes, two people that are heterozygous have a 1/4 chance of producing a child with sickle cell disease, but they can have 10 kids in reality and none of them have sickle cell disease, or they can have 10 kids and all of them have sickle cell disease. It's all a random chance. However, as we get our sample size big enough, as we get near 100,000 or 10,000 births with this genotype, you're getting closer and closer to 25%. So it's the same thing here, right? With this small sample, what we can say is probably it's big N, but it could be she could be a carrier. So what's the probability that either of the children between Ralph and Alice, right? They have two children, which I didn't draw here, so I should have, but we don't know what it is. What's the probability that their children will be blind? What's the probability that either of their children will be carriers? Explain your answer. So to answer it correctly, you have to do two different scenarios. One in which if Alice is big N, then you get this. If Alice is big N, then you have this situation. So if Alice is big N, then you have a 0% chance of children being blind, because she doesn't have a little n to give. And then there's a 50% chance for Ralph to be a carrier, right? Because Ralph could be big N. We don't know. But there's a 50% chance that Ralph could be a carrier. So that means 50% of the children could be carriers. There's a 50% chance that the children are carriers, right? Because he can get the half of his kids, 50% chance he gets big N, and 50% that he gets little n. Now, if Alice is big N, little n, then you have another situation. You have Ralph, we know, is at 1/2 a chance of giving little n. The chances of Alice giving a little n if she's a carrier is 1/2. So that's a total of 1/4 of a chance that the kids are carriers. All right. What are the chances the kids are affected? Well, there's a 1/4 chance that Alice can give it. There'd be 1/2 a chance that Ralph can give it, and that gives you an 1/8 of a chance that the kids are blind. All right, so that's how you'd answer that question, to be completely honest. And it's long. This one was actually harder than this one. I should have made this one mandatory and this one extra credit, but that's the way it was. All right, so that's the end of the genetic section. Again, I want to remind you, if you want that 20% boost, rewrite your answers in your own words that match the right answer, and you'll get your 20% boost. Now let's go on to protein synthesis in the next video.